Good morning, students. The topic for today's presentation is cDNA library. Students, we have already discussed as to what a DNA library is. The second type of DNA library that we'll discuss today is cDNA library, which contains the complementary DNA synthesized from mRNA molecules in the cell. A cDNA library is defined as a collection of cDNA clones that are copies of all of different mRNA found in a cell. Because working with mRNA is difficult, an enzyme called reverse transcriptase is used, which produces a DNA copy of each mRNA strand. Now we'll discuss some of the differences between the cDNA and the mRNA. The cDNA is a double-stranded molecule while mRNA is single-stranded. The cDNA is stable whereas mRNA is highly unstable. cDNA is easy to manipulate while mRNA is quite difficult to manipulate. Next, the cDNA needs to be transcribed into RNA to make a protein, whereas mRNA can directly make a protein. The construction of a cDNA library involves the following steps. The first step is extraction of RNA, followed by isolation of mRNA, synthesis of first strand of cDNA, followed by synthesis of its second strand. Next step involves treatment of cDNA ends, followed by cloning into a suitable vector. Now we'll discuss the steps of cDNA library preparation one by one. The first step, as we said, is extraction of RNA. This step is very important. It should have optimal disruption of cells, effective denaturation of nucleoprotein complexes, maximum inhibition of RNases, and effective purification of RNA from DNA and proteins. The second step involves isolation of mRNA. In this particular step, the total RNA which has been extracted in the first step is passed down a column of oligo-DT cellulose. As we very well know that the eukaryotic mRNA has poly-A tail at its 3' end, as we can also see in the diagram, it binds to the oligo-DT matrix. Now, when this mixture of cytoplasmic RNAs is put under hybridization conditions, only the eukaryotic mRNA binds to oligo-DT primer, whereas the other RNA, that is rRNA and tRNA, are washed off. Later on, the column is eluted under the influence of a low salt buffer. Purified mRNA is obtained. The third step in the preparation of cDNA library is first strand synthesis of cDNA from mRNA. There is a need of synthesis of cDNA from mRNA because mRNA is a highly unstable molecule, so it becomes very difficult to deal with mRNA. The enzyme used in this step is reverse transcriptase, and also an oligo-DT primer is required, which starts the synthesis of first strand of cDNA with the help of 4 deoxynucleotide triphosphates and magnesium ions, as can be seen in the diagram. At the end of this step, we get one strand of mRNA and one strand of cDNA, so it is called as cDNA mRNA hybrid. Now we move to the second strand synthesis of cDNA by self-priming method. As we saw in the last diagram that a mRNA cDNA hybrid is obtained. Now the mRNA is removed from the single-stranded cDNA mRNA hybrid either by boiling or by alkali treatment. After this step, it is seen that the first strand has the tendency to transiently fold on itself, forming hairpin loop, resulting in self-priming of second strand. This leads to the formation of the double-stranded complementary DNA as seen in the diagram. Later on, the loop is cleaved by another enzyme, that is S1 nuclease, to allow the insertion into a cloning vector. The next method for the synthesis of second strand of cDNA is replacement synthesis. This method was given by Okayama and Burge in the year 1982. The enzymes used are DNA polymerase 1 and RNase H. RNase H actually cleaves the RNA component of a DNA RNA hybrid, leaving short oligoribonucleotides attached to the cDNA. This can also be seen in the diagram. Now, these short stretches of oligoribonucleotides serve as primers for the polymerase enzyme to synthesize the second strand of cDNA. In the end, the newly synthesized strands of cDNA are joined by the process of ligation with the help of DNA ligase enzyme. The next method for the synthesis of second strand of cDNA is homopolymer tailing. In this particular method, an enzyme, terminal transferase, along with DCTPs is used to add homopolymer tails of DC residues to the 3' end. Thereafter, in the next step of diagram, we can see that mRNA is removed by alkali treatment. In the next step, oligo-DG is used as a primer, and then the second strand synthesis of cDNA is accomplished. The next, that is the fifth step in the preparation of cDNA library, is the treatment of cDNA ends. The cDNA that are synthesized have blunt ends. Now, if we have to insert them into various vectors, they should have sticky ends. So this can be done with the help of linkers, adapters, or by homopolymer tailing. After the treatment of the cDNA ends, the cDNA is ligated into a suitable vector to generate clones. Post ligation, the clones are transformed into a suitable host to get colonies. With this, the cDNA library is constructed. After the construction of cDNA library, it is screened for identification of cDNA clone of interest. Thank you.